Hello, everyone. My name is Paul Kirika from the Botany Department of the East African Herbarium at the National Museums of Kenya. I'll give an overview of the fungal research in Kenya, specifically the research carried out at the East African Herbarium. First, I would want to thank the organizers of this meeting for inviting me to give a presentation. And I will start by giving a brief introduction of what fungi are. And fungi are small filamentous organisms comprised of masses of thread-like hyphae, which constitute the body of the mycelia of the fungus. And they can exist as single-celled, for example, yeast, or as multicellular body comprising of interconnected network of mycelia. They are found everywhere in all ecosystems and in all habitats, including aquatic and both aquatic and terrestrial habitats. And they have some important characteristics. They digest food externally by excreting enzymes into the environment and absorbing the dissolved organic matter back into their cells. So they basically live in their food. And most have cell walls composed of chitin, a substance which is found in the exoskeletons of insects and shells of crabs and lobsters. They also store their food reserves as glycogen and lipids, and not starch as in plants. Genetically, they are distinct, and their DNA indicate that they form an independent kingdom, which is more closely related to the animal kingdom than the plant kingdom. Fungi are heterotrophic and therefore cannot be able to make their own food. So they get their nutrition through various means, including living as saprops, where they break down dead organic matter. And in the process also, they cause a lot of destruction to food, leather, and timber, and therefore causing huge economic losses. Others live as parasites, where they feed on living organisms. And in this process, they also cause diseases. So most of the bad pathogens for both plants and animals are parasitic fungi. Others live in symbiotic relationships. For example, the mycorrhiza fungi, which form symbiotic relationships with the green plants, and also the lichen forming fungi, which form symbiotic relationships with algae or cyanobacteria. Others live as predators, where they trap small microorganisms such as amoebas and nematodes. They do so by secreting sticky substances over the surface of their hypha, and therefore causing any animal that touches any portion of the mycelia to stick firmly to the hyphae. And then the hyphae grows and penetrates into the tissues of these animals and release digestive enzymes. Fungi are important ecologically. They sustain life on Earth by decomposing organic matter, breaking it down into simple molecules and releasing carbon, oxygen, and nitrogen, and even phosphorus, release it back into the soil and to the atmosphere. So they're important in nutrient recycling. Mycorrhizal fungi also provide key benefits to terrestrial ecosystems by enhancing the uptake of nutrients and water by plants, and also are important in the formation of soils, and also they are important in carbon sequestration. They act as sinks for plant carbon. Since time immemorial, man has always used fungi for his needs, the production of foods and beverages. Over 350 species of edible mushrooms are collected world over for food, and some species of fungi are also used in the production of, for example, meat substitutes, in the ripening of cheese, and bread making, and also production of food colors. And alcoholic beverages, for example, all the beers, wines, spirits, other liquors like Busa, Moratina, and Kalovo, they are all produced through fermentation by yeast. The uses include enhancing plant growth and in the production of medicines and drugs. For example, penicillin, which is used for the treatment of bacterial infections, and cyclosporine, which is an important drug in organ transplant. Fungi are also used in the production of hallucinogens. In fact, more than 200 species are believed to be hallucinogenic, and majority of which are found in the genus Psilocybin. Other application of fungi to human use include production of enzymes, which are used in the making of washing detergents, processing of leather, and paper manufacturing. Today, fungomycelia is also used in making of substitutes for leather in building, and also in the production of biofuels, where they are used to break down agricultural waste such as maize and sugar straw, which is fermented using then fermented using yeast to produce ethanol. They are also used in bioremediation, where they are known to degrade PCB chemicals in soil and polluted waters. One of the most important components of research is the documentation and preservation of the fungal diversity. Kenya is known to have a rich fungal diversity, and they are studied through collecting, visiting different regions, and collecting specimens and data that is associated with those specimens. And as you can see, these are just some of the some of the fungal diversity found in Kenya. And so to study fungi, scientists collect herbarium specimens and data associated with the with this collection. And these are the vouchers that are used in taxonomic research, in ecological and biochemical, in ecological research and biochemical analysis. And they form reference material for the accurate identification of fungi, while also providing a reliable data on the distribution of taxa. And they are also the core material upon which application of scientific names are based. These are mainly the type specimens. Therefore, give a permanent record of species at a particular time and provide information that are used in the production of fungal checklists, and also important information on monitoring. Another area of research is the establishment of fungal cultures, both mushrooms and mycorrhizal fungi, for use in agriculture, forestry, and in the restoration of degraded habitats, and also in promoting mushroom farming. Good quality mushroom spawn is produced for farmers so that they can have a healthy crop, and therefore able to have a good product. The other area of research is the wild edible mushrooms, which this is spearheaded by our colleague, Dr. Joyce Jeffwa, who recently got a grant through NRF. It's out of the realization that mushroom has a great potential to diversify diet and contribute to food and nutritional security in this country, and also increase job opportunities and improve livelihood. So the project is focused on studying the taxonomic diversity of indigenous edible mushrooms, and also determining the distribution, ecology, and production capacity of these indigenous edible mushrooms, while also evaluating the different methods of preserving the genetic materials, and also trying to devise appropriate ex situ and in situ conservation approaches for the indigenous edible mushrooms, and also determining their safety through carrying out nutritional and toxicity analysis. The other area of research is the study of mycorrhizal fungi, which are important in the maintenance of plant species communities, habitats, and ecosystems. And these involve the study of vascular mycorrhizal fungi and ectomycorrhiza. And it's important to point out that in the conservation of rare and endemic plant species, the study of the associated mycorrhizal fungi is of utmost importance. To conclude my presentation, I would say that the fungal diversity in Kenya is not very well documented, and data on fungi for most areas is lacking. And this gives opportunities for fungal research in taxonomy, evolution, ecology, physiology, genetics, and even chemistry. And that is the end of my presentation. Thank you very much, and I would welcome any questions or any comments. Thank you.